Hey again, everybody. This is where we picked up last. We left off with hemolytic disease of the newborn, but I want to talk now about white blood cells and wrap this chapter up, hopefully, if not in this video, in the next one. Oh my gosh, I don't know what just happened. Ignore that. So white blood cells, WBCs, as I've been calling them, leukocytes. Leuko translates from white, or I guess it's Greek, into white, and then cytes is cell. So, or cyte without an S is cell. All right, so these guys have a nucleus. If you're talking about each particular cell, this could be misleading. It sounds like they may have more than one, and some of them, in fact, actually look like they have more than one, but that's not the case. They do have a single nucleus plus other organelles, but based on the type of white blood cell, and I will show you pictures of them, you'll see that they have what are called multi-lobed nuclei or nucleus. Okay, they do lack hemoglobin because their job is not to bring in oxygen and to get rid of CO2. They have many, many more functions, whether it's defending us from bacteria or viruses, whether it's removing cell debris and toxins from an infection, or cells that have gone through what's it called when they rupture? Apoptosis, which is our planned cell death. And then they attack abnormal cells or damaged cells. So this could be something that's infected with a virus, it's become cancerous, or a cell that is bruised because somebody bumped something and now they have a bruise. So these guys have way more functions than just delivering oxygen and removing CO2. And we're going to see more of that. And then the other thing I was going to say is these are the cells when you see on TV where like they found a blood sample, the white blood cells here, where they actually get the DNA from the criminal or the victim or whoever, but not the red blood cells. So keep that in mind. That's the second time I've mentioned that. All right, so most white blood cells are going to be found in our connective tissues. They are, as well as our organs, for our lymphatic organs, which we will get to shortly after the chapter on cardiovascular and blood. I can't remember the chapter number. All right, so a small percentage of the white blood cells circulate in blood, so about 5,000 to 10,000 per microliter. And if you haven't seen this term yet, I hope that's not the case, but it will say like 5,000 per microliter. And this letter here is the Greek mu, like meow. And if you could think of it as like a little m that's not really quite drawn out, but it's supposed to be a little u. So microliter. All right. So when they talk about white blood cells and how they migrate out of the bloodstream, they're talking about moving into the tissues that have either pathogens present, been damaged, or anything like that. And they do, if you guys want to see something cool, Google diapedesis. I didn't spell it right. Pedesis. There we go. Diapedes. I think there's an A in there somewhere. No, there's A. Okay. But Google this, and I'll try to put it in the PowerPoint so when you guys click on it, you guys can see it. But anyways, so here's my vein, not an artist. You guys know that by now. Here's my white blood cell. It gets a signal from a damage over some kind of damage or a cut over here. It gets a chemical signal, and then it literally rearranges its entire cytoskeleton, the skeleton of the cell, and actually fits through little endothelial cells, goes right through it after it rearranges. It's pretty insane. And you don't have to worry about the ICAMs that use them. That's basically the braking system for these guys. But this would be what's called positive chemotaxis. It's towards the injury site. And they're literally following these little messages. And I like to have my students think of them as a text message. Hey, I'm over here, or I'm over here. I just checked in over here. Some of the white blood cells are phagocytic, so they will eat cells or bacteria or viruses or damaged cells, et cetera, et cetera. And some are not. Okay. So here's the different types. And again, these are from lightest to darkest. Neutrophils, eosinophils, basophils. I put them darkest to lightest and remember Ben. And then there's also the never let monkeys eat bananas. So never let monkeys eat, bananas. Eat. Okay. But I don't like this one because it mixes up the granulocytes, these guys, with the or agranulocytes, these guys. I don't really prefer that one. I like Ben, and then I just remember monocytes. And remember also these monocytes will become macrophages when they grow up. So I'm just going to put max right there. That's really, I'm going to put max, macrophages as an abbreviation. They grow up and they're going to be highly phagocytic. Okay. So here are the pictures. Please, please, please recognize these without their names being right here. So if you see them on the test or in your lab, you know what they look like. Now, if you're asked if you can differentiate if this is a T lymphocyte or a B lymphocyte, the answer is no. So don't fall on that trap. True or false? You can differentiate between T or B lymphocyte with this image. No, you cannot. The answer is false. And it could be worded differently, and you could say true. So it could say you cannot differentiate, and you'd say true. Okay. All right. Monocyte has this guy. Looks like he kind of like, it's not the greatest Pac-Man picture, but he kind of looks like he's gotten punched right here. It's still one nucleus, but it's called an indented nucleus, right? Basophils, these are the darkest staining ones. And we're going to get into their actual functions slide by slide. Eosinophils and neutrophils. I know it looks crazy, but this is all one nucleus right here. This is all one nucleus. Hence the term multi-lobed nuclei or nucleus. There's not multiple nuclei, so that's another question. And this guy he's attached right here. Right there. There's a single nucleus, and he just happens to be attached right here. But they're multi-lobed. So if you had an ear that had 4 lobes, you'd still have one ear. It would just have multiple lobes. And that's how the nuclei in each of these cells looks like. All right, so neutrophils. These are, they're also called this, but I've never called them that. But they have polymorphs. This is the majority. So then the highest percentage present. If you just knew that, you'd be fine. You don't have to know this number. But they're the highest percentage present. And they're also the first on the scene. So if you get a big old fat zit, or I hate those things, or you have a paper cut or whatever, they're going to be the first on the scene. And they're usually responsible for the pus, right? The white pus or whatever you have in the injury. All right. So they do stain the lightest. So they have a pale granules. They're still granulocytes. They still release granules. These granules have lysosome, which hopefully you remember from Bio 1, will attack and break down things. They also respond mostly to bacteria. So bactericidal means bacterial killing, like homicidal means people killing. So bactericidal or virucidal means viral killing, right? So these guys are bacterial killing compounds. They're granules, kill bacteria with the lysozymes and the other compounds they carry. Do you need to memorize all of them? No. Do you need to know they're activated based on bacteria and then the highest ones present and the lightest staining ones? Absolutely. Okay, so here's a nice picture. Again, multi-lobed nucleus, still one nucleus. All right, very highly active. They are phagocytic, meaning they can engulf another cell. If you want to have some fun, you can Google S. aureus. And I don't know if neutrophil is in the title, but S. aureus and phagocyte. And you'll find it's like a 30-second video. It's pretty fun. All right. So not only do they attack these bacteria, but they'll break them down and make sure that they can't attack you anymore. Does this work for all bacteria? No. But it does help with our immune system. So, and again, we're going to see these again in the immune system. So don't be like, I don't need to know this. Okay. So again, like I said, the dead neutrophils contribute to pus in an injury. They only stay alive and active in the blood for about 10 hours or less. And they do release prostaglandins and leukotrienes, which we're going to see the role in vasocongestion and vasodilation. when we get to the inflammation part with coagulation. Not coagulation, sorry. Shenanigans. Clot formation. Okay, eosinophils. These guys, you don't have to worry about eosinophils. That's just how they stain them. They're a far smaller percentage. Okay, they will eat bacteria, protozoans, and cellular debris. The basophils also will increase the presence and attract more neutrophils. Right? And notice it's neutrophil, not oh, not this guy. Drat. This is, I'm sorry, this is for basophils. So we'll just, that's not correct. When we go, we'll skip ahead and come back. All right, so basophils are going to increase and attract neutrophils. Okay, and notice there's no E here. They're not phials or phils. Neutrophils, as well as eosinophils. Get attracted and increase. So basophils are doing that. We'll come back. So, okay. They do like to attack, when we're talking about eosinophils, they like to attack large parasites. So think of intestinal worms, which is gross, I know, but that's what they like to attack. Okay, so they're going to release nitric oxide, which is NO. It's a gas, and that is pretty harmful for these. Unfortunately, you're talking about a little tiny cell versus a ginormous worm. So they don't always win. So, yeah, I got that right. Okay. And then there are cytotoxic enzymes, which means cell toxic enzymes. So they're, they can kill a cell that is, they can, whatever they release. I'm sorry, guys, brain fart. They release toxins that kill cells. They also, eosinophils are also, oh, here it is right here, activated during allergens. So when you take an antihistamine, you can reduce the presence of eosinophils. And there's been a correlation that you guys probably aren't interested in, but the lack of our exposure to parasitic worms in our country and in first world countries around the world, where you can walk around barefoot most of the time and not pick something up, the lack of our exposure to parasites is believed to be correlated strongly with our increased sensitivity and our increased allergies, so our increased allergic reactions. So we're going to see enzymes that get released by the eosinophils, reducing inflammation, right? That the inflammation specifically caused by mast cells and neutrophils. So these guys will come in, they roll in after the neutrophils. And again, this is wrong. It's on the next slide. They roll in after the neutrophils and the mast cells, which we'll see mast cells in the immune system. But they roll in afterwards, and they slow, the eosinophils will lower the inflammation a little bit. Boop. Here's your basophils. Again, they increase and recruit neutrophils to the site of injury, and eosinophils. These are a small percentage of what's present. These guys do that capillary endothelium crossing towards damaged tissue. Basophils release histamine, which allows the blood vessels to get bigger, dilate, right? They get bigger. And heparin, which prevents the clotting of the blood. So this will allow other neutrophils and eosinophils, other granulocytes and agranulocytes to come in and do their job and keep you healthy. Okay. When we discovered heparin, and how it prevents clotting, it wasn't long after that we were like, oh, we can put this on the inside of blood tubes when we do collections and prevent clotting in the blood, so that the blood samples can last longer when the labs are trying to, when the labs are trying to look at the blood work and see what's going on with the person. All right, monocytes. These guys, again, remember, they grow up, they become macrophages. They'll hang out in the bloodstream for up to 24 hours. When they get into the peripheral tissues, they're no longer called monocytes. They're now called macrophages, which I just call max. All right. So this is where they aggressively phagocytize different pathogens. And I wanted to say with monocytes, I meant to say this is going to be, it could be large pathogens or dead cells. Okay. I did say that. And then they do release chemicals again that attract other phagocytes and cells, fibroblasts, which we have not talked about, but we will when we get to immune system. Okay, so monocytes I covered. Dead cells. Oh, these are also going to be the antigen presenting cells when we get to the immune system. So when you see me use APC in immune system, I'm talking about antigen presenting cells. Once the monocytes are activated, the macrophages are then referred to as antigen presenting cells. So they phagocytize something, they break it down inside, and they take a little piece of it and express it on the outside for your immune system to recognize. Like a little signal, hey, look at me, I'm over here. This is what you need to pay attention to. What I wanted to say with basophils, again, I did mention the neutrophils and eosinophils. This is histamine, again, increases the blood flow. Another way to say dilates blood vessels is to increase vasocongestion. Okay, which means the blood gets more congested. Okay, so eosinophils talked about nitric oxide, but I wanted to mention. I did mention parasitic worms. So, what worms here? Just so you guys haven't. And then I feel like I didn't. Did mention bacteria. Where's the cancer cell one? Why can't I remember? All right. Number. Thinking. One of them attacks cancer cells. I'm going to go ahead and say macrophages do it. And when I remember, I will post it. All right, so these guys are a little bit larger, as you can see, than red blood cells. They pretty high up there with circulation. They're continuously going around the bloodstream in that connective tissue matrix, and monitoring stuff. And they do this mostly in the lymphatic system, hence lymphocytes, right? So they're gonna. I already said connective tissues for blood, but the connective tissues other than blood, so the lymph is what they're talking about. And this is specific defense system. So we'll get into that when we get into that. All right. So 3 classes for lymphocytes. You've got your T cells, your B cells, and your natural killer NK cells. These guys are, if a antigen presenting cell comes in and says, hey, I found this piece of bad bacteria, I'm going to show it to you. It's cell mediated. So this antigen presenting cell, which is a macrophage but was a monocyte, comes and says, hey, T cell, I got something, wake up, let's go. And it's going to attack the foreign cells that are not lymphocytes. The B cells, when they say humoral immunity, they're referring to fluid immunity. So it could be plasma, it could be body blood, it could be lymphatic fluids, but it's going to be fluids, not tissues. And these is not turned on by another cell. They're differentiated into plasma cells, where we get our antibodies as well as our gamma globulin that I mentioned at the beginning of this chapter. That's an ugly gamma, sorry. All right, so but these guys differentiate into plasma cells. This helps with our immune memory, right? So again, we're going to see all of this, like both in immunity as well as here. So you might as well knock it out of the ballpark. Okay, so natural killer cells are going to say, hey, what's not supposed to be here? And I'm going to destroy it. And this can be cancer cells, viruses, bacteria, all kinds of stuff. But the cancer cell ones really bother me now, guys. I know it's in the freaking book, but I don't see it right now. You guys are like really tired of listening to me wait. So annoying. Okay, I thought I wrote it down. Guess not. All right, so differential white blood population. If you take this, we can count what type of white blood cells are present. Are they Ben? Are they lymphocytes? Are they monocytes? So basophil, eosinophil, neutrophil, lymphocyte, monocyte. We can go there and say, okay, this you have possibly parasitic worm infection. It's just allergies. You have a virus. You have bacterial infection. La la. We can figure that out. And these numbers mean something for us. If we were in lab, you guys would be doing these on a slide under a microscope, but we're not doing that. And I haven't found it in the activities for mastering. Okay, so leukopenia, yeah. It means you have low white blood cells, so your immune system is not reacting as it should. Leukocytosis, you have high white blood cells, which means something is definitely happening. And again, we would look and see which of these are elevated and what is exactly going on. And leukemia itself is speaking about cancer of the white blood cells, and it's extreme leukocytosis, so extremely high numbers of white blood cells. And we're making some progress on leukemia, but not as fast as I would like. All right. The production of white blood cells, which I'm just going to say white blood cell production, but make sure you recognize leukopiosis. And then understanding when we get to the figures we're coming up, I want you guys to be able to tell me where the progenitor cells are. And then I'm just going to move to the figure because it'll be easier. We do develop them again, remember, from bone marrow. There'll be some lymphatic stem cells, right? Or some hemopoietic stem cells, right? And then we get B cells, natural killer cells. Then others from our lymphatic tissue, they'll give us cells from the thymus, the spleen, lymphatic nodes, and lymph nodes. And they produce our lymphocytes yeah. And then, if you guys could definitely remember, T cells are produced in the thymus. Okay. So, B cells are from bone marrow. All right. So remember, B cells are from bone marrow, whereas T cells right here are from the thymus. And when I'm in person, I usually say T cells are from the thymus, right? Just so people can remember and maybe have a little goofy laugh at me. That's fine. So T cells are produced in the thymus. Colony stimulating factors. We're not going to really worry about multi-stimulating CSF. And remember, it's when they say CSF here, it's not cerebral spinal fluid. It's colony stimulating factors. I told you guys earlier, the G is granulocyte. The M is monocyte. I think I've already covered all that. Okay, so this figure, please do know that you've got hemopoietic stem cells. Again, it's going to be from the red bone marrow. Blah. You could just say bone marrow. That's fine. Or don't pick white bone marrow. Right. So red bone marrow. And then you've got myeloid, which means to come from the bone, and lymphoid, which means lymphatic. So you've got your colony stimulating factor, right? And then I would like you guys to know that EPO, erythropoietin, plays a role in the first couple of steps before you get to the erythroblast stage. The next thing I want you guys to know is that before it becomes a reticulocyte, so to go from an erythroblast to a reticulocyte, it has to not only inject the nucleus, but also has to make HB, the hemoglobin we talked about earlier. Okay, so that's what I would like you to know from this one. The platelets are from a megakaryocyte. That's always kind of fun to know. We rarely get to see these guys in under the microscope in white blood cell or CBC count. All right, so granulocyte. Oh, no. Okay. And then monocyte stimulating factors. Again, if you could know those. I did ask you to know the progenitor cells. So we need to know that all of our, whether it's a red blood cell or we're talking about the white blood, the plasma is not over there, white blood cell, these guys come from myeloid, which means from bone. Lymphatic is going to give us our lymphocyte, right? And then the monocyte is included. It also comes from the myeloid. So, okay. So only one, the lymphocyte, comes from the lymphoid stem cell. The rest of them all come from the myeloid stem cells, right? So you don't need to know all the steps from myeloblast to myelocyte to band cells. What I would like you again to know is to know this row of items. Other than I've also indicated what else I want you to know. But basophils are the darkest, eosinophils are the medium color, and neutrophils are the light color. Monocytes become macrophages, and lymphocytes are both T and B. So just keep that in mind. So with this example, this lineage we looked at here, because it comes from the red bone marrow, these would be your B lymphocytes. And our T lymphocytes would come from the thymus, okay? So you don't need to freak out about that. But if you're looking at a blood smear on your test or a quiz or something like that, keep in mind, you can't differentiate based on that slide if it's a B lymphocyte or if it's a T lymphocyte, okay? I think this is just where they break it down and make it look a little bit nicer. That's exactly what this is. All right. Platelets or thrombocytes. I prefer the name platelets myself, but whatever. These guys are, again, fractionated or fragmented cells. They are going to be highly involved in the clotting system. They do circulate for over a week. They can be removed by phagocytes. Usually this happens in the spleen. So when a surgeon's like, hey, I'm going to take your spleen out, like, hey, can you patch it? Because I kind of need it. You don't have to, whatever. I'm just kind of kidding around. And this is based off of like the numbers. This is kind of a large range, but 150K to 500K per microliter of blood. So again, that's a mu liter of blood. That's an L. You can't read my handwriting. 1/3 of your platelets are stored in vascular organs, again, like your spleen. And they get mobilized when there's a crisis of circulation in your body. To get them released, you're gonna, for the functions of platelets, there's gonna be clotting chemicals that come out. We're gonna temporarily patch these vessels, and then the clot's gonna shrink, and the scar is gonna replace it. All right, so production of platelets is thrombocytopiosis. Piosis. It does occur in red bone marrow. Red bone marrow, just like we saw from the picture, the diagram earlier. Megakaryocytes are giant cells, like we saw. And as they break apart, they actually shed these little parts that become platelets. And when they shed them, we'll use them they'll be around in the blood, but they're going to become they're part of the cytoplasm. If you want to go back to that really big picture, this guy. So as they get shed, these little parts right here that become the platelets are part of the cytoplasm, and they look to me like little tiny flatworms, but whatever. So megakaryocytes, they start out giant and they end up giving us little tiny platelets. All right, so a couple of hormones that are involved is going to be thrombopoietin or TPO, interleukin 6, and colony stimulating factor. And it's going to get really complicated when we look at the blood clotting phase. But for homeostasis, we have to maintain. If you get cut, you need to be able to you need to stop that bleeding. And cessation is a fancy word of saying stop. So smoking cessation classes help people stop smoking. Cessation of bleeding helps the body to stop bleeding. So there's 3 different phases: the vascular phase, which is the most common phase, and I'll show you an image in a minute. The platelet phase, which is temporary, but it gives us the feedback to get or excuse me, it's going to be a positive feedback. So it gives us a positive feedback to get us into our body's cells into the coagulation phase and shut down the bleeding. So when we get to the coagulation phase, keep in mind that fibrinogen, the structure fibrinogen, is going to be turned into fibrin. And you don't need to freak out about all the enzymes involved. The fibrin will then be turned into a clot. After this clot retracts, as this clot retracts, scar tissue will come in and replace it. All right, vascular phase again, it's going to be the most common. A small cut will trigger this, but you'll basically get the smooth muscle, and I'm not the greatest artist, fibers of the smooth muscle portions of the vessels, which is obviously not consciously controlled. These guys will contract. Where are my pictures? Okay, so if you, I oh my god, see paper cut freaks me out. All right, so it gets injured, and this is what I'm trying to say, the vascular spasm that's most common. They contract, it's smooth muscle, so it's not conscious, it's unconscious. Not dilation, contraction. It can say lasts about 30 minutes. This is the point where if there's a cut, you want to make sure that you, instead of wiping the injury, you want to blot. Okay, to get if there was any bacteria in there, blot it first. Unless this is like a spurting Monty Python style injury, you want to blot this injury to gently get the bacteria out. So it's blot, not wipe. And as soon as a little bit of blood or a decent amount of blood has come out, then you can actually put pressure on it. Okay, and allow this to move into the next phase, right? Which would be your platelet phase, the positive feedback phase, and then the next one, which is coagulation and clot retraction. Well, clot retraction is the last one, but all right. So peptide hormones called endothelins are going to be involved. ADP is going to help release and do this positive feedback cycle. Again, the smooth muscle contraction, it's not something we think about. It's not something we don't think about digesting our food, right? That all happens based on chemical signaling that's going on in our body. And if you need to revisit some AMP1 materials, please do. If you need me to explain it some more, please let me know. All right, so endothelial plasma membranes, the ends of them become sticky. They'll seal off and tear, or seal off a cut to prevent blood from flowing. And I totally messed up. Paper cut. Sorry. Okay. So they get smaller. The platelet phase, so this is the second part, right? And this is the positive feedback that we saw things getting upregulated for in the last phase, right? They'll attach to the exposed surfaces. They then will stick to each other using these little pseudopods. Okay. It generally is pretty rapidly happening, depending on like how many vitamins you have in your blood, where the cut actually occurred. And this forms the platelet plug that can close small breaks. If you have an arterial bleed, that's not going to happen. You're going to have to put pressure on that thing and go get that thing stitched up or cauterized or whatever. All right, so platelet phase continued. Again, these factors are going to activate it. So thromboxane A2, serotonin, other clotting factors. Platelet-derived growth factor, which if you've watched the first set of lectures in the endocrine system, this is another one of those positive feedback. There's so few of them that this is one of those few positive feedback mechanisms in our body. When they say calcium, they mean Ca2+. But there's so few of them. So platelet-derived growth factor is a positive feedback system in our body. My face hurts from talking so much. All right, so here are all the clotting factors that get released. So here's where the cut was, but you can see we're already in the platelet phase, which is, again, temporary before we get into the coagulation phase. So you guys get these ones, and the smooth muscle is starting to contract and pull things together, so that less and less blood is lost. I wanted to say something else about this. This phase takes a bit longer than the platelet phase, but that's, you guys can read, I'm sure. All right. So factors that will limit the growth of the platelet plug are prostacyclin, and that prevents aggregation. So there's inhibitory compounds released by white blood cells, breaking down enzymes that run around and break down ADP. And then there's serotonin, which has a negative feedback loop. All right, so the last phase, coagulation phase or blood clotting, it says begin 30 seconds or more after the injury. Again, that depends on the size of the injury, but you've got procoagulants. These guys, coagulants mean to plug up, right? So procoagulants would mean to help plug up, right? There's 11 different proteins. Do you need to know them all? No, but calcium 2 would be a good idea to know. If it's called a proenzyme, it's like a provirus, but it's not active yet. It has to be converted to an active enzyme to direct reactions. And then there's going to be 3 different pathways. So we're going to see, again, extrinsic, intrinsic, and common. We're going to see this again when we get to immune system. And I'm going to stop this here and see you guys in the next video.